Listed like us minor, but here fall on us.
No, they called me and they're like, yeah, I can't come fix it tonight, but we'll come fix it.
Like for our community, if it's something small, we have to fix something major.
They have no water going to their toilet, like none of them have showers since yesterday morning. And now it's like 80 degrees there.
So then we have to fix our air conditioning. So, and I'm not sure. Other building across from yeah, set up and got the water set up. Everyone we have set up is. Ours is not far, ours is a whole house. One bed, one bath.
So it's like, not like a big basement, like it's a dirt basement, so we can't,
The other setup was the alpine stuff down there too, it would also cause. It can do. We have like a little, it's gonna be like outside factory uses like door, doesn't have to lock and dry. And then we'll put like my bin, stuff on that way, like, and we'll put up like a curtain. Okay, I was about to say, what? You think you get an apartment?
I don't think he was happy about that either, but I'll figure.
Well, good afternoon, everyone. How's everyone doing? Great. Isn't that how all your professors start class? How's everyone doing? Hopefully you're doing well. I mean, you're in the home stretch, right? You only this is the last week of classes, and you got final exams next week, papers, projects, and things like that. So we're nearing, I don't know, I think we're about halfway through chapter 16, so we're on track to finish this one up on Friday, by Friday then. I was really impressed with your presentations yesterday. You did such a good job there, and each one of you, I want to say that how proud I am of the work that you've done. You accomplished the goal of characterizing, identifying your microorganism. And you may have noticed on that sheet that you used yesterday for your presentation that you ended with a fun fact, right? That's something that I always appreciate. So today I'm going to return the favor. At the end of class today, I will share a fun fact with you. Okay, so something to look forward to, right? All right, before we get to the fun fact, let's remind ourselves about Coase postulates here. I can't remember who was the one that worked with the bacterium that was actually first identified by Coke. Was it you, Mallory? Okay, good. So recall that Coke developed these steps here, the postulates, in order to help us to understand the causal agent of infectious diseases. So how do we know that this bacterium causes that disease? Is by applying these steps here. So so we're going to run through these to remind ourselves, but then we're going to look at and learn about what are some of the problems associated with these steps, or why can't solve all known cases here. So step number one, remember, it is to build causation versus correlation by identifying the microorganism in every case of the disease. And then the next couple of steps involve isolating that microorganism, injecting it or exposing it to a susceptible host, and then observing to make sure that that host will actually develop the signs and symptoms of that disease. Let's remind ourselves, what do we mean by a sign? What do we mean by a symptom? This is where you shine.
Signs are measurable, symptoms are objective.
Yes, signs are measurable, symptoms are objective, right? Or they're hard to quantify, right? Or subjective, yeah. All right, good. And then the last step here, the idea is to re-isolate that organism, like a bacterium, grow it as a pure culture, separate from everything else, and then do the staining, the biochemical tests, the DNA sequencing to confirm that the organism that was isolated, that was causing the signs and symptoms of the disease, is in fact the same organism that was injected or exposed to that host. All right, so let's move forward. So, sounds good, right? 4 steps, 1, 2, 3, 4. This slide here lists some of the problems or limitations associated with Coase postulates. So let's look at, so we can understand what some of these are. First one is that some organisms cannot be grown in culture, separate from their host in petri dishes, on media, in the laboratory. So there's actually most fungi, we're unable, we don't know how to grow them in the laboratory outside of their natural environments. We just haven't figured out, what are the nutritional requirements in order to do that. So that's where we need you. I know we live in a very high-tech, sophisticated world, but there's a lot of problems out there. I think you can see that there's a lot of problems out there in the world that need solutions for. Some of these problems are really big. Some of these problems are less so. So, like, here's one problem that is not, I wouldn't say it's like, hey, everyone's going to die if we can't figure out how to grow some fungi, but the idea is that there's so much out there that you can still contribute to better our understanding of. Okay, next point here. Infected individuals do not always show signs or symptoms of that disease. Remember, there's a term for those individuals who harbor that pathogen that don't show any evidence that they have that pathogen. Do you remember what we call those individuals? Carriers. That's right, carriers. So think back to Koch's postulates. You know, you're exposing or injecting a host, and you're expecting to see a disease, right? But in the case of a carrier, you don't. So that stuff doesn't quite work well. Third point is that some of these diseases can be caused by, you know, not just one, but multiple organisms. So again, when you're trying to build that case of causation versus correlation, ideally you want to see one disease caused by one pathogen. But things are more complicated when you could actually have multiple other microorganisms causing essentially that same disease. And then the last point here, suitable animal hosts are not always available. We don't like to test using human subjects. So that last point is really important there, especially like in cases like when scientists were trying to figure out, what's the causal agent of AIDS? Well, today we know AIDS is caused by what virus? HIV, right? The human immunodeficiency virus. Well, if we were to carry out Koch's postulates with HIV, we would have to, knowingly inject human test subjects with that virus to see if they would come down with AIDS. We don't like to do that. We don't consider that to be a good thing or ethical. So that's another problem using Koch's postulates. Right? Okay. Any questions about these kind of limitations or problems associated with Koch's postulates? Okay. So, in order to try and resolve some of these problems, scientists have come up with, sort of like alternative ways of determining that a microorganism can cause disease, a particular disease. And that is by taking a genetic or a molecular biology approach and looking for genes that are associated with virulence. So remember, that's like the degree of pathogenicity. So it could be something like the ability to make a capsule, like that exterior slime layer that will protect bacteria from being destroyed by phagocytosis. It could be something like the production of a toxin that will actually damage host cells and inflict signs and symptoms of diseases that way. So it could be a variety of different genes that are responsible or contribute to the ability of that microorganism to cause disease. So the idea here is that, well, we can identify that gene as playing an important role by disrupting its function through mutation. There's a lot of different ways of doing that. One simple way is to expose those organisms to mutagens. Things that cause genetic changes, things that we know cause genetic mutations. Like there's a bunch of chemicals out there that we know cause mutations. UV light, we know causes genetic damage and mutations. So it's that kind of stuff where we can expose these bacteria. We can determine if mutations have happened by using DNA sequencing. And then the idea here is that if that gene does play an important role in the ability to cause disease, then by mutating or disrupting it, we should see less virulence, or the loss of pathogenicity. And likewise, with the ability of molecular biology, we can restore, we could actually do a transformation experiment, mobilize a normal version of that gene, put it into a bacterial strain that has the mutation, and then the idea is that by recombination, you can swap out the mutated gene with a normal functioning copy, and then you should be able to restore virulence as well as pathogenicity. So there's other ways around the issues surrounding host postulates. All right, any questions about this one here? Okay, let's move on here. All right, so next we're going to learn a little bit more about how the organisms can cause disease. So that's the referred to as the mechanisms. Pathogenesis refers to the ability or the creation of disease. Okay, so these microorganisms can cause disease in a number of different ways. How can they infect? So first example is with the production of toxins. So we're actually going to end this course on Friday by looking at two kinds of toxins, two general categories of toxins that we call endo and exotoxins. So we'll learn more about those types there. So sometimes it's not the microbe itself, but rather the products of the microbe that's actually responsible for the diseases that we see. We already talked a little bit about the importance of colonization. So in order to cause disease, we typically need to see a certain number of the microbe must be present. And do you remember what we call that number? Sometimes it's abbreviated as an ID. What word starts with I? What word starts with D? Okay, so two words. I'm looking for two words. First word starts with I. Second word starts with D. First word is related to disease. Infectious, and then the second word, think about numbers. The number of pathogens required to cause disease, the infectious. One word, 4 letters, starts with D. Dose. Thank you. The infectious dose. Okay, so another for disease that happen, colonization. During colonization, you have multiplication, cell division in order to give rise to that critical number needed to cause infection or infectious dose. Then we have invasion of host tissues and the avoidance of host defenses, and we'll see examples of that. So remember that we have the innate and adaptive defenses. There's a lot of stuff going on there. You have those antibodies, you got the cytokines, you got the complement system, you got the phagocytes. So there's multiple ways in which the pathogen, in order to cause disease, needs to prevent those things from preventing its multiplication. When we're talking about invasion of host tissues, we're talking about the ability to penetrate our exterior skin, penetrate through our mucous membranes, get inside of our bodies, get inside of cells. Okay. So let's learn a little bit more about these things here. All right, so this statement in red mentions that typically we see a balance between the pathogen and its host. If you think about it, if you have a really lethal pathogen, what would happen to the host, to the host species? Think about it. If there was a really deadly, let's say there's a really deadly virus out there. Let's say it's a human virus, 100% lethality. What would happen to the human species? We would get wiped out, right? Every human would die. Okay. Well, what then would happen to the virus? It would be eliminated too, because it would require humans in order to replicate. So if it's super deadly, it would not continue on. Does that make sense? Yeah, because the host would be completely wiped out. So for any pathogen, in order to persist, it needs the ability to replicate, but not so severe that it completely wipes out its host. Okay. Now the host is not going to just, we're not just going to stand still, right? So remember that we have our adaptive immune system, and after primary infection, how does our body respond after that? It creates special cells that can recall that microorganism. Cells we call, what kind of cells? Memory cells. That's right. We create memory cells that will then be able to fight off the second time that we encounter that particular pathogen. So because we have that adaptive response, that means the pathogen, in order to be able to reinfect, needs to up its game here. So that means through mutations, improve its ability to avoid our host defenses, maybe make more lethal toxins or other kind of substances that in order for it to survive. And then, remember that we're not just static organisms, we adapt. So the next time we encounter something more severe, we will adapt, our bodies will adapt, and then generate even more memory cells the next time around. So there's kind of like the seesaw balance going on between host and its pathogen. Both are adapting to each other, right? So that's what they mean by the balanced pathogenicity between host and its pathogen. When you were young, did you ever read Alice in Wonderland? Yes, okay. There's one of the characters is called the Red Queen, and it's been a while since I've read that, but part of the storyline was, there's a lot of crazy stuff described in there, but part of the storyline was, I believe it was the Red Queen was running, continuously running, but never moving, never getting ahead, never falling back, always running. So that was used as a way of understanding and actually describing this balance and pathogenicity. So this is actually referred to as the Red Queen hypothesis. The Red Queen hypothesis, that as much as the pathogen is trying to cause disease, the host is continually evolving to adapt so that neither one gets the upper hand. They're both moving, changing, and yet getting nowhere. It kind of describes life. All right, let's move on here. So next we're going to learn about how these microorganisms can, some of the different steps involved in causing an infection. So Brady, I believe in your presentation you had a microbe where you described just like a halo. Yeah. Right? You had where it's like a little halo around the microbe, the bacterium, and that halo was caused by Peelle, also known as Fimbriae. Peelle and Fimbriae, which are these, there's like these spaghetti-like strings extending out from that microorganisms. The Peelle and Fimbriae are designed for attachment to help that bacterium to adhere to surfaces. It could be inanimate objects like a rock, or it could be a living thing like what we see here, which represents the membrane of a cell. So it's actually attaching to proteins extending outward from the plasma membrane of a host cell. So that's what we mean by adherence. In order for infection to occur, the microbe has to attach to its host cell. So, recall that, same thing is happening with viruses. Recall that when we learned about the bacteriophages, they have to land on their host cell. That's like the first step. Okay, then we have colonization. So remember, the microbe begins to multiply. And in order to multiply, it has to, it's going to encounter, a variety of different host defenses that it needs to overcome. So one of those is the acquisition of iron. So you've probably heard you need to eat some foods that have iron because that's an important mineral for your metabolism, for binding of oxygen. Well, microbes need iron too for metabolism. They need it for oxidation reduction reactions, for transfer of electrons. And there's kind of a scarcity of iron, so the bacteria actually needs to try and acquire it from your body. And so it has these special siderophores that it uses to bind in order to access and use your iron for its own metabolism. So it needs to compete with you for iron. Okay, the second point here, or not the second, but another bullet point here is that the microbe needs to avoid this over here, the IgA. Ig is an abbreviation for immunoglobulin. Immunoglobulin. There's another name for an immunoglobulin, a more common name. Anyone know what it is? Immunoglobulin, what is that? Think immune system. Think adaptive immune system. Antibody, there you go. The immunoglobulin is an antibody. So it's made up of protein. It's those Y-shaped structures. Remember that they can bind, the ends of the arms can bind to antigens. There's like 5 different classes. So this is the class A. That's what the A is for. Okay, so the microbe's going to encounter antibodies, and in order to try and avoid them, some of these microbes make proteases. We've talked a little bit about prefixes, suffixes. What does the suffix here? The ASE, what does that tell you about that word right there? A protease. Yes. It is an enzyme, that's right. Yeah, your vocab is improving by the miles today, isn't it? So the IgA protease is an enzyme that the bacteria makes to break down your antibodies, so the antibodies won't work like they're supposed to. Andy, where did you learn that?
I don't know, it's like I just heard it before.
Okay, there you go. I wasn't sure if you've taken biochemistry or something like that. No? Okay. Last point here is that the microbe must compete with your normal microbiota. Now you have a lot of microbes. I don't know if you realized or noticed, like yesterday during those student presentations, how many of you were actually describing organisms that are part of your microbiota. So that's great that you have so many different kind of microbes there. But that also means that the pathogens have to have have a lot of competition there to deal with, for space, for nutrients, and so on. Okay, so the bacterium has to adhere, has to start multiplying, so it reaches, increasing its numbers to that beyond the infectious dose required to cause disease, needs to avoid host defenses, and then one way you can avoid host defenses is by injecting molecules into host cells using this device right here. So here's the host cell down on the bottom here. Okay, so pretend this is a human cell. Pretend this is like the cell wall of a bacterium, and then look at here. There's this needle-like. It's like a syringe here, a needle-like syringe that it uses in order to pump effector molecules, these are chemicals, into the host cell. So then the host cell, in order to block or prevent that host cell from mounting a full immune response. Okay, so it's kind of like, let me give you an analogy. It's like someone injecting you with drugs to prevent you from responding. So this is what the bacterium is doing at the microscopic level. Pretty insidious, right? Okay, and then what? Your innate system, what you have from birth, you have a very effective barrier established, in both your skin and in your mucous membrane. And those are barriers that the microbe, needs to cross in order to access additional cells in your body. So let's look at the first case here. In order to penetrate your skin, remember most of these bacteria, they don't have the ability to bore through your epidermis there. So they're going to enter through cuts or wounds, or use other organisms like fleas, ticks, in order to have them serve as vectors to breach your skin. So I mentioned this before, but it's important whenever you have an open cut or wound, make sure you clean it, but then cover it up with a Band-Aid so that you don't, to prevent exposure of that wound to microorganisms. I often forget about these fleas and other kind of biting insects, but fun fact, what is the deadliest animal in North America? It's what? Mosquito? The mosquito. They're actually insects. Yeah, they're different kinds of insects because they harbor these pathogens. So I know when I first said that, you're probably thinking, bear? Lion? Tigers? Oh my! But no, they are the tiny insects, the flying insects, the biting ones, like the flea. There's other pathogens that can actually enter into your cells by inducing them to perform endocytosis. So, that's the process that phagocytes use when they engulf organisms that are on the outside and then bring them inside. And so one thing that microbes can do is that they can induce changes in the plasma membrane of a host cell and cause those membranes to, basically form an open pore. And on the outside, this is kind of like, they call it a ruffle. Maybe think of it as like a funnel, creating an opening, through which that bacterium here can, go through that plasma membrane and enter inside of that cell. Here's just kind of a visual of endocytosis. So, for example, here, try to imagine this brown rectangle here is one of your cells. Here's our microorganism at the top. We don't see any ruffling, but the idea is that would be the ruffling would take place at the surface here. But we do see endocytosis. So the membrane would fold around that organism, and then that membrane vesicle would internalize right here. So this is a way that pathogens use in order to enter into a cell. So I kind of like fooling it to perform endocytosis. Another way that pathogens can avoid host defenses is by hiding within cells. So in red, we have a list of some of our host defenses, like the complement proteins. Remember that complement proteins can bind to these pathogens and by opsonization promote phagocytosis. Well, that binding happens outside of cells. Likewise, antibodies can bind to pathogens, but that also happens outside of cells. So if the pathogen is inside of the cell, then those processes won't happen. So that's what they mean by hiding in a cell, so that complement proteins, antibodies cannot bind to them. Okay, and then related to that, both complement proteins and antibodies can facilitate opsonization, leading to phagocytosis. So this slide mentions that the bacterium could actually degrade. It has a peptidase that will break down one of the complement proteins. So if that occurs, then you don't have opsonization. And then another possibility is the bacterium will actually kill the phagocytes. So if you don't have opsonization, if you don't have the phagocytes, then you're not going to get destruction of the microbe. So you're not going to get phagocytosis. Okay, one more slide before the fun fact here. So another possibility here is avoiding binding with the antibodies. So, because antibodies serve as a tool for opsonization. So this is like what would normally happen here. But then look here on the right side of the slide. This bacterium here has special receptors on it. It will bind to the bottom of the antibody instead of the antibody binding to with its arms. So this will not lead to opsonization. So this is one way that the bacterium can escape being targeted for phagocytosis. All right. Any questions about any of this so far? Okay, let me show you a real quick video here. Let's see here. Let me go back to our Blackboard site. Okay, a quick video. Are you guys familiar with Jeopardy? Okay. All right. I don't know what the. Okay, I know that was a short clip there, but that was actually from last night's episode of Jeopardy. And a number of you know that I went to the University of Wisconsin last week to give a talk. I gave a talk on my book. My book is called The Soul of DNA. So my book was actually an answer on Jeopardy last night. So that's the fun fact. That's how we're going to have the class here today.
That's awesome, man.